A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Sallallahu alayhi wasallam. I mostly start my lectures with this slide just to emphasize the fact that what we are understanding or remembering at the moment will guide us, will help us to evaluate different medical facts in future. We are starting our topic of enzyme, and inshallah we will conclude it in 7 to 8 lectures. The lecture outline for today's lecture is Introduction of Enzymes and Nomenclature of Enzymes. The reference book, as you know, for the medical biochemistry are Harper, Lippincott and Mushtaq. In Harper latest edition, which is 31st edition, you may find the course of enzyme in 3 lectures starting from the 3 chapters starting from the chapter 7 on page 156, and in a Lippincott 7th edition, which is the latest edition, on page 53 in a chapter number 5. In Essential of Medical Biochemistry volume 1 on page 139, chapter number 6. What are enzymes? Enzymes are biological catalysts. So before discussing anything about enzyme, it's better to have a brief look on the feature of catalyst. Catalyst is a Greek word meaning to dissolve. Means dissolve any issue, dissolve any matter. It's just like a troubleshooter. So in a chemistry, a catalyst is a substance that can be added to a reaction to increase the reaction rate without getting consumed in that process. So the basic feature of catalyst that it increases the rate of reaction, it converts the substrates into products quickly but without getting consumed. It remains permanent at the end of the reaction and can be utilized repeatedly for that particular reaction. However, it modifies during that reaction, and this fact we will elaborate in a future lectures. There are millions and trillions of spontaneous reactions happening in our body to maintain the normal physiology of the body, means the homeostasis. So metabolism is sum of all chemical reaction involved in maintaining the living state of cells and organisms. Metabolism grossly divided into the catabolism and anabolism. So after brief discussion of catalyst and metabolism, now we come on the main topic, which is enzymes. By definition, protein catalyst that increases the rate of reactions without being changed in overall process are called enzymes. So enzymes are basically proteins in nature which increases the rate of reaction and does not consume in that particular reaction. Enzymes thus direct all the metabolic events happening in our body. So enzymes can convert a single substrate into single product A into B, like isomerases and transferases we will see in the lecture of our classification of enzymes. A single substrate into multiple products like hydrolases or lyases. A multiple substrate like A plus B into C, like ligases, or multiple substrates into multiple products. We will see in our future discussion as we go on with this topic of our enzymes. How the enzyme do all these miracles or these magical things? It increases the rate of reaction by decreasing the activation energy. If we focus, if we consider A is being converted into the B, and without enzymes we can you can appreciate the blue line, and with enzyme you can appreciate the red line. It decreases the activation energy, and how it do it we will see it in our future lectures. So all enzymes are mostly protein in nature. However, some RNAs like ribozymes can act like enzymes. There is a famous saying, however, it's not correct, that all enzymes are protein in nature, while the all proteins not are not enzymes. Mostly the globular proteins are enzyme in nature, but there are few enzymes which are not protein in nature, that are RNAs or ribosomes. Enzymes make the life on earth possible. Without enzyme, life is not possible on this earth, in this world. How they do it? They do it by participating in the breakdown of nutrients to supply energy or chemical building blocks. Whatever we eat, either carbohydrates or proteins or whatever we take through our respiratory system like oxygen, it converts into the energy biomolecules and basic building blocks, and body do it by the help of enzymes. By assembling these building blocks into proteins, DNAs, membranes, cells and tissues, and by harnessing of energy to the power of the cells for its motility purpose, for its neuronal functions, or for its muscular contraction. It is the basically these are the enzyme on which our basis of essentials or non-essential biomolecules depends. Like essential amino acids are those amino acids which we must have to take in our diet. Why? Because the enzymes needed to produce those amino acids are not present in our body. So there are thousands of enzymes in the living organism, showing great diversity in their action. So to differentiate them, their naming is very important. So each enzyme is assigned by the two names according to the Lippincott, which is first name is a recommended name or convenient for everyday use. The second naming system is systematic name, which is used when an enzyme must be identified without any ambiguity. Basically, these two systems are recommended name or systematic names depends on the three basic principles. The first principle or first basis is depends on the substrate which are being converted into product. Like we simply attaches a suffix ase with that substrate, and name that enzyme. Like sucrose, we attach a suffix ase with the sucrose, and then the name of enzyme become sucrases. For lactose, we can correlate lactose with the enzymes lactases, urea with the ureases. The second basis for the nomenclature of enzyme is the kind of reaction they are catalyzing. If in a substrate water is being added and breaking the larger molecule into a smaller molecule, we can uh, name that enzyme, particular enzyme, hydrolases or dehydrogenases. Mean we will see it in the redox reaction. The systematic name basically is the combination of these two facts. Enzymes can be made of enzyme name can be made of two parts. The one part or word indicating the substrate, and second word indicating the reaction or the action that particular enzyme is doing, and the second part end with the suffix ase. For example, lactate dehydrogenase, in which in this systematic name of this enzyme, lactase is a substrate which is being converted into pyruvate by transferring its two hydride atoms and being converted into the pyruvate. So, lactate first word indicating that its substrate, and dehydrogenases indicating the uh, about the reaction of this enzymes. International Union of Biochemistry IUB developed a systematic names nomenclature in which each enzyme has unique name and code number that identify the type of reaction catalyzed and substrate involved in that reaction. For example, the IUB name of hexokinase is ATP D-hexose 6 phosphotransferases, and its EC number, which is enzyme code or enzyme commission number, is 2.7.1.1. In this EC number, the class, the 2 indicates the class which is transferases, the 7 indicate the subclass which is transfer of phosphoryl group, the 1 indicate the subclass alcohol is the phosphoryl acceptor, while the hexose 6 indicates that the alcohol phosphorylated is on carbon number 6 of hexose. There are few limitations of this nomenclature, which is some enzymes retain their original name which give no hint of associated enzyme reactions, like trypsin or pepsin. We cannot judge anything from these names, like pepsin. We cannot correlate it with hydrolases, transferases, or any substrate it's being acted upon. So, there are few limitations of this nomenclature system. So, enzymes are very critical biomolecule of our body, as enzyme allow reactions in the body that would be impossible otherwise. Almost all the reaction, spontaneous reaction happening in our body, either in forward direction or reverse direction, we need enzymes for these spontaneous reaction. That is the end of the today's lecture. From today's lecture, we will be able to know about the nomenclature system of the enzymes and few basic things about the enzymes. In the next lecture, inshallah, we will discuss the classification of enzymes.